Right. So, somebody's joining us from Istanbul. Very warm welcome from Istanbul. Thank you for joining us. So, my name's Kim Morgan. I'm the Outreach and Communications Coordinator here at UCL Biochemical Engineering. And after asking those questions, I'd like to launch a poll. So, the first question is, what level of study are you interested in or would be interested in? So we're trying to work out whether our audience is mostly prospective degree or postgraduate students. So I'm going to launch that poll and you'll see it appear in your little poll thing. It should have popped up. If you could just click on that and let us know so we can work out what percentage of our audience is at what prospective level. Previous sessions have been very mixed. We've had some that are more undergraduate, more postgraduate. Interestingly, today it's almost very evenly matched between prospective undergraduate and prospective postgraduates. Just slightly edging ahead for prospective degree students. Very interesting. Thank you very much for that. So I'm gonna say that in this session we will be pitching this at sort of A-level, IBAT level, possibly undergrad level. We just want to make it clear that if you find any of this bit beyond your level, if you're at school or college, if you come and do a degree with us, we will bring you to the level where you understand this. So any of the learning or any of the principles that we discuss here, any of the terminology, our modules are structured in such a way that you will be brought to this level. So don't feel like, I don't understand this, I couldn't study with you. What we do is we will bring you up to this level and beyond. So I just want to reassure you there. The next question is whether you actually have an offer to study. Somebody's actually pointed out in the chat and let us know that they are a career advisor. Thank you very much for joining us. It's really interesting to have you on the webinar. But it's great to have we're also hoping that with people beyond prospective students are actually interested in what we're doing. So it's great to have you here. Interestingly, at the moment, it looks like the majority of the people don't have an offer to study with us. But then quite a few do have an offer to study with us. Well, a very warm welcome. Thank you for joining us if you have an offer to study, especially because this is kind of a little bit of a taster about who we are and what we do. And one of the reasons why we do these sessions is to really help people understand what they might be studying here. Thank you very much for that. Really appreciate you letting us know. And it's very interesting that most of the people don't have an offer to study here. So we're thinking maybe prospective undergraduate students. Thank you very much for that. So the way this session is going to work is I'm going to explain how the setup works, and then I'm going to hand over to Steffi. I will be here in the background throughout the whole of the session. But if you have any technical problems, say if you can't hear us clearly, if there's any kind of problem with the slides of the presentation, you can let us know using the chat facility. You'll see that within the Zoom controls you have the option to chat. You can message me, and myself and Steffi will see that. Somebody's already posted a question. I'll come to that now because you can actually post questions at any point in this session. But if you could put them in the Q&A box, it's really helpful because you will find that some of these will be answered as you go through. So if you ask a question, you might find that later on Steffi does address that. Also, we find that sometimes people ask similar types of questions, so we can bunch them up. The Q&A function means that we can see which questions we've answered and we can see which ones are pending and we can group them up. So please feel free to ask the questions at any point. We'll answer them towards the end, but use the Q&A box. Finally, this session is being recorded. We'll send a link afterwards, but it takes us a few weeks to process the recordings because we need to add subtitles to them all, and it's quite time consuming to add subtitles. I think that's it. Finally, just to say, towards the after Steffi's done her presentation, I'll talk about some of the other events we've got coming up. Because if you are interested in studying a degree with us, we actually have some open day sessions taking place, one tomorrow and one on Friday. So you can come and join us online and talk to us in detail about degrees. But now I'm going to hand over to Steffi. I'll turn my camera off, but I'll be here in the background. Okay, thank you.
Thank you very much, Kim. I'm going to share my slides now. Right. Hopefully you can see my screen. Good. Good morning, everybody, and welcome, and thank you for joining this early in the morning. And it's a pleasure for me to talk to you today about bioengineering of vaccines, and also some manufacturing. But I'm particularly interested in kind of discovery and bioengineering aspects of vaccines, as you will see during the presentation. So what will we do in this session? I will talk to you about half an hour, and then we will, you can post your questions in the meantime, and I will answer them after my presentation. So I will talk a little bit about what biochemical engineering is, what our department is like, and what we offer, and then what the role is of a biochemical engineer within kind of vaccine development and manufacturing, because we're talking about vaccines today. So you'll see how biochemical engineers are important in developing and manufacturing vaccines. So we will talk about why we need different types of vaccines, how vaccines are designed, and also how vaccines are manufactured. On the right-hand side, actually, in this image, you can see the UCL main building, and below this, you can see our department, Department of Biochemical Engineering, which is very nice. Let me use the pointer as well. So we see our building. So first of all, I'd like to tell you a little bit about myself, so you know who's talking to you today and where I'm coming from. And perhaps it's interesting for you to see kind of how I got into the biochemical engineering department. So Kim, by the way, your mic is still on. All right. So I'm from Germany, as you can probably hear. Hopefully my accent is not too strong. So I did my undergraduate degree in Germany and in Jena. And I actually did a nutritional sciences degree, but with a lot of biochemistry, lectures and so on, which I really enjoyed. And I became very interested in fundamental research, and I wanted to become a biochemist. And I applied in the UK because I knew the UK was very strong in research, and I wanted to do my PhD in the UK. So I couldn't find a PhD position initially, so I took up a research technician post at UCL many years ago, 20 years ago now. And I was working in developmental biology, working on a wound healing product. And, we started forming like a little spin-out company with my supervisor and my colleagues. And then I went on to do my PhD in biochemistry at Queen Mary University, also in London. And I was working on vitamin B12 biosynthesis, so very, very much biochemistry. And after I graduated, I did a number of postdoctoral research positions. So this is after your PhD, you work as a researcher in the lab on several projects. And during this time, I became very interested in studying bacterial compartments. So these are protein structures that are found in bacteria, and they fulfill some kind of function within the bacteria. They help bacteria grow, and in fact, they do also help some pathogenic bacteria to become more pathogenic. So I studied those, and I started engineering those kind of structures for the biotechnology applications. And in this time, I also had my first child. You can see her here. She's now a teenager. And then I wanted to become an independent researcher, so I applied for funding, and this is called a fellowship application. So you apply for your own funding to form your own group and to follow your passion in research. And I did that next, and then had twins in the meantime. So we had 3 babies at the time, or small children, so it was a very busy time, but luckily I still had time to pursue my passions, and I continued working on these micro compartments and the engineering. And then this was kind of a board towards becoming, like a full-time academic. So I applied for academic positions and in the biochemical engineering department at UCL, and I got that a lecturer position in the department about 5 years ago, so 2017. And So since then I've been teaching on several undergraduate courses. For instance, I lead a vaccine course for our undergraduate students on vaccine bioprocessing. I also teach our postgraduate students, so our PhD and NhD students. For those of you who are interested in postgraduate studies, I teach basic research skills that any research student would want to have when they start their research project. I've also become involved, I told you I'm a synthetic kind of biologist, biochemist, but I started becoming involved in vaccine research as part of the VaxHub, and I will tell you about that in a minute. And I became the leader for the UCL iGEM team, which is a synthetic biology team that does work on real world problems using synthetic biology, so engineering biology to make, to improve and make certain products. Anyway, so just a couple of facts about our department. We are the largest academic bioprocess group in the world. So our department is excellent in teaching and research all around biochemical engineering. We have about 24 to 25 academic members of staff, quite a good number of undergraduate students, so 250 in total. So we take in about 50, 60 students per year across various programs that we offer. We have a high number of doctoral researchers, so we have lots of researchers and postdoctoral researchers, so that researchers that go on to continue research after their PhD. We are very multidisciplinary, and just trying to move this window out of the way. So we also have a lot of collaborations with industry because many of our research projects are industry driven. So they're driven by some industry projects. So we do some fundamental research, but with industry input. A number of spin-out companies have formed from the department and from the research that has been performed in the department. And we have two major hubs, the VaxHub and the Future Manufacturing Research Hub, that carries out extensive research in kind of future manufacturing of biopharmaceuticals and vaccines. And in terms of the kind of work we do, kind of the research we do is our teaching. So if you're interested in undergraduate degrees, our teaching is very much driven by our research and our kind of research ideas. So we're trying to take our research into the teaching, and as you see, we have a lot of researchers in the department. So we work on industrial biotechnology, kind of three major themes: industrial biotechnology, biopharmaceuticals, and cell and gene therapy. And vaccines kind of fall under biopharmaceuticals. So we work on recombinant vaccines, for instance. I'll talk about that later, proteins or genes, and also complex chemicals. And in terms of cell and gene therapy, you had another session on cell and gene therapy. Some of you may have attended it. Work on kind of stem cell therapies for various diseases and other types of therapies. Underpinning all of that is our kind of research expertise, very interdisciplinary. Some of our researchers look at high throughput processes, so designing a bioprocess to make a certain product. How do we make, let's say, a vaccine in a fermenter? How do we purify that product? How can we scale that up to like more industry scale? Because we work from bench scale to larger scale towards kind of industry scale. And then we use also, and this is more like my area, synthetic biology tools to optimize proteins and plasmids, so DNA, and also engineer cells to make more product or make certain products. We also have kind of analytical technologies that we develop, because a lot of our products, for instance, particularly vaccines and other biopharmaceuticals, they need very stringent controls. So you need to understand very, very clearly what is it that you made. So you need to be able to analyze the product. And then we have a number of researchers that work on decisional tools. They model a certain process, how a product is made more efficiently, or they do an analysis of the life cycle of a product, or they use AI, so artificial intelligence, in the process of making products. So in terms of the VaxHub, I was going to tell you a little bit more about that, I promised you. So the VaxHub is the Future Vaccine Manufacturing Research Hub. This is a big kind of research consortium where it's so to speak a hub. It has a very ambitious program and is led by 2 directors. One is Martina Micheletti, who's leading the hub from the UCL side, and the other director is Professor Dame Sarah Gilbert. You may have heard about Sarah Gilbert. She developed the Oxford AstraZeneca vaccine in 2020, and they are co-leading the hub, and they raised a lot of money, 9 million, to do this research work, innovative work. To develop vaccines, to scale up vaccines and manufacture vaccines with particular benefit to low and middle income countries. So the initial idea was to kind of develop better ways of making vaccines for those kind of countries. The work that had been undertaken in the VaxHub, because a majority of that work is now completed, has really helped the rapid development and the manufacturing of the Oxford AstraZeneca vaccine that you know about, which is called ChAdOx. And we talk about that as well in a minute. And now the vision of the hub going forward is to generate the next generation vaccines, generate for a rapid response. If we have another pandemic, we need to be able to rapidly respond, and also for innovation of vaccines. But I was going to tell you also a little bit about iGEM, just very shortly, because if you're an undergraduate, potential undergraduate student in the future, we run this synthetic biology team every second year now. So every 2 years, we're putting a team together of undergraduate students, and we have postgraduate students helping with the project. And we're trying to solve some problems like, plastic degradation, because there's obviously a big problem with plastic. Or we're looking into this year, we're thinking about looking into biomaterials for building, so kind of more sustainable building materials, and other kind of projects. And every year, this competition runs worldwide, and it culminates in kind of a big jamboree at the end of the year, where all the teams from the world come together and present their project ideas. So this is called iGEM, for International Genetically Engineered Machines. And just to show you, we have had a team in 2019 who's worked on a protein drug delivery particle that was meant to target breast cancer, and this is why we're wearing all these T-shirts here. And they went to the jamboree, and we got a lot of prizes, and we got a nice gold medal there. If you're interested, I've posted a paper here. We published the work as well. So let's go over to vaccines now, telling you a bit more about vaccines now. So a little bit of history. You probably heard about smallpox as part of school education, but the first kind of very prominent vaccine was the smallpox vaccine against the smallpox virus. And we talked about the VaxHub and Sarah Gilbert, who developed the COVID vaccine. Actually, Sarah incidentally works in the Jenner Institute in Oxford, and the Jenner Institute is named after Edward Jenner. So he was a physician and a scientist in 18th century, and he actually found out that smallpox was very widely prevalent at the time, around 1790, with many people dying, 400,000 or so people dying every year, people going blind. It was a big problem. So Edward Jenner found that a young dairy maid called Sarah Nelmes, it was known at the time that dairy maids were somehow not affected by smallpox as much as others. So he found this young dairymaid, Sarah, who had a fresh cowpox lesion, so on her hands and on her arms. You see this picture. Apologies, it's not a nice picture, but smallpox causes these kind of lesions, and so does cowpox. Yeah, so they are similar pox. And he used, because he thought, oh, these dairy maids, they must be somehow immune against this kind of smallpox virus. So he used the cowpox lesion from Sarah's hand, and he inoculated an 8-year-old boy called James Phipps, and the boy developed some mild fever and some discomfort. But after about 10 years, he felt better, and he then inoculated the boy again after some time, in July actually of the same year, with a matter of this time smallpox, yeah, from a lesion from smallpox. And the boy did not develop any disease, and so Jenner concluded that there was protection from this cowpox lesion that he was initially giving to the boy. So the boy was immune, essentially. So this was kind of the first vaccine. And at the time, it was so revolutionary. He was trying to publish this, Jenner was trying to publish it at the Royal Society. People were not taking him seriously. They're saying, no, this is gross and you shouldn't do this. But it was so successful that the first vaccine developed, and eventually smallpox was eradicated. And in the 1980s or 1980, the World Health Assembly announced that we are now free from smallpox. And the Latin word for cow is vacca, and cowpox is vaccinia. So Jenner actually decided, after this success, to call his new procedure of putting, this lesion from one person to another. He called this vaccination. So this is where the terminology is coming from. So, what is a vaccine? Each pathogen, and this could be a pathogen, this could be bacteria, virus, or or something else, has several kind of subparts, the way it's composed. And these are usually unique things to the specific pathogen. And these cause normally a disease. So for instance, a bacterium has a lot of molecules on the outside, kind of sugar molecules that often cause, they cause disease. And so this subpart, for instance, the sugar molecule on the outside of a bacterium, of a pathogenic bacterium, causes the formation of antibodies in your body, in our bodies. And so this part of the pathogen, of the bacterium, is called the antigen. So the antigen is a specific thing to a bacterium or a virus that causes the formation of antibodies in our bodies. And a vaccine can be any kind of cellular component from a bacterium or a virus or something else, and a preparation of that kind of part, and can then be used as an antigen. So we can give a part of a bacteria or virus to a patient and use that as an antigen. And the antigen will then stimulate the immune response in our bodies. And on the right-hand side, you can see here, for instance, a measles virus, or a flu virus, or chickenpox virus, and they all look a little bit different on the surface, right? They have different molecules, and our bodies make different antibodies against those different molecules on the surface, which is described. And if something new comes into your body, you haven't got any antibodies, you've got to make them against this kind of virus. So we have now, luckily, many vaccines against many different types of diseases. You can see a list of diseases on the right-hand side of the screen here that we have vaccines for. In the news recently has been cervical cancer vaccine, HPV, because HPV has been developed now many years ago and has been highly successful in preventing cervical cancer in women and girls and women. So we're using this very successfully now. And there's also lots of other diseases that we can now treat very successfully. And these are, particularly kind of childhood immunizations that you probably had as a child, measles and so on. You can see some examples here on the left-hand side of some viruses and bacteria that we now have vaccines for. So each of those vaccines has had their own process developed, and these processes had to be developed by researchers, scientists, like yourselves in the future, and like biochemical engineers. So this is where biochemical engineers come in again. And there are some challenges to vaccine development and manufacturing. We will see in a minute that vaccines are actually quite high to make, high cost to make. So they're quite expensive. They take a long time to be made. In terms of how we make them, is quite different because it depends on the disease you're trying to try to make the vaccine for, depends on the kind of the pathogen and what the pathogen looks like. And they require very stringent analytical analysis, because we need to be absolutely sure that the vaccine is safe and it's what we think it is, because we have to fulfill high safety standards, because we give vaccines to healthy individuals and particularly to children. I said, we just looked at this very long list of, childhood diseases like rubella and typhoid and pneumonia and so on. So we need to make sure that, we gave this to kids, that the vaccines are very, very safe. So I kind of want you to appreciate how long it takes normally, kind of in non-pandemic times, to make a vaccine. So if we look at the vaccine development process, it will take many years, 10 to 15 years, or even longer to make a vaccine. Some vaccines take more than 15 years to make. And they're very expensive to make. They cost about 200 to 500 million US dollars per vaccine, per type of vaccine to develop. Why is that? So this is because, first of all, it takes years to come up with a good candidate. So in the lab, the researchers in the biochemical engineering department, in my group and other groups in the Jenner Institute and so on, and other institutions, we spent many years looking at, good vaccine candidates. Which protein should we pick from the virus? Should we take the whole virus? Should we just take a little bit from the virus or from the bacteria and so on? How can we make this? How can we produce then the vaccine candidate? Then we need to do preclinical trials. So these essentially, when you test your candidate on animals like mice or other animals, and that can take several years, because you need to work out, is it safe? On what dose do I need to take? How many milligrams per milliliter do I need to give to a mouse or to to some other animals? And then after this, you go into the different phases of clinical trials where you give your vaccine to humans. So we test for safety in humans in a phase 1 clinical trial. That takes months. Then we test in a phase 2 trial, again for safety. That can take up to years. And then we test for safety and efficacy again, and that can take up to tens of years. And then we market the vaccines. That can take a few months. And then you do long-term surveillance, like with the COVID, with the pandemic, the vaccine got rolled out. We take the vaccine, and then it's not finished, right? We're still doing the surveillance. We're still looking, okay, do people, report any side effects? How does the vaccine work? And so on and so on. It keeps going on and on. So I'm showing you the same timeline again, this time like with these colored boxes. And I'm showing you this because I want to show you what happened during the pandemic. So this is our traditional timeline again, many, many years. So our initial design of the vaccine, then we need to develop a process. So how do we make the vaccine? How do we manufacture it? This is where the biochemical engineers come in again, at the design and at the process development and at the manufacturing. And then the clinical trials have to be done, and we have our kind of, 3 phases, phase 1, 2, 3 that I've just shown you, sorry. And then the regulators are very, very stringent. So after you've done all your phases of trials, you need to show all the data to the regulator, say, look, here's my data, everything looks safe and it's efficacious, it works. Then the regulator says, okay, it's good, you can go ahead, and then you do the large scale production and you distribute to different countries, to low middle income countries and so on. And this takes a long time, and it's always step by step, one after the other. And investment is very difficult because companies were quite reluctant in the past to invest in, each of those steps, it takes investment, and it can take many years again, because, you might run out of money after the phase 1 trial and you get more investment to do the other trials. So what happened in the pandemic was we had an accelerated timeline, meaning that we now have kind of shortened this timeline, but not because we're jumping any of those steps. It's because we were able to do some of those steps in parallel. The initial development was kind of the same, and kind of the toxicology studies in animals is the same. But then for the kind of clinical trial studies, these were done in parallel for some of those kind of vaccine candidates, because we had a lot of knowledge of the candidates beforehand already, and they had been already in clinical trials by the time, by the time the COVID pandemic hit us, and we were able to go forward with them, because we had already some data. And some of these trials were done in parallel, but they were done with a huge number of people, and they were done very thoroughly, the same as before. But also, and that's really important, there was a lot of investment, a lot of money went into making COVID vaccines and into kind of making large scale production, where you make the vaccine already kind of at risk, because we didn't know, perhaps the manufacturers didn't know at the time, will the vaccine actually hit the market? So they invested and they make all these big batches, millions, billions of batches at risk. But this was important because we had to treat the entire world. So this is why the vaccine timeline was shrinking to about 12 months. So from the time when the kind of the sequence of the spike protein was released, the DNA sequence, to the point of having the first vaccines available, it took 12 months. So huge speeding up of the timeline. And also the regulator finally was looking at all this data, whilst the data was being produced, the clinical trial data. They didn't wait to the end. They were looking at everything and doing, working with the clinical trial data very quickly. So I would like to tell you a little bit about the different types of vaccines. You saw the different diseases we're treating. What are the different types? We have lots of different types available now. For instance, we can make live attenuated vaccines, which means we can take a whole virus. It's a live virus, but it's actually not as it's not as dangerous to the body. It does not cause the same effect. It just causes an immune response, and your immune system will be able to deal with it. So it's a kind of a weakened virus. Then we have inactivated viruses. They are essentially whole viruses, but they're dead. They have been killed. And these two are our traditional vaccines. So we've seen them for many, many years, for kind of measles and so on. We've had them around for many years. So we have to have very specific vaccine manufacturing processes for these as well to make them, and they require high safety, so the people that manufacture those don't get infected, and so on and so on. Later on, we started making these parts of the virus where we just take, for instance, if you imagine this was a coronavirus, we're just taking the spike protein, which is a little protein on the surface, and just produce that spike protein from the virus, nothing else. And this is kind of a much safer vaccine in a way in terms of production, but it needs addition of some ingredients to cause a good immune response, but it's very effective. So these kind of vaccines are very effective. Then we have these polysaccharide vaccines that are kind of sitting, sugar molecules on the outside of bacteria that we can use. And we have these kind of virus-like particles where we also could use a virus, but we don't use the virus with the DNA or RNA inside. So we're removing all the genetic material from inside, so the virus cannot multiply within your body. It's just kind of the outside, the capsid of the virus, and that will cause a really nice and really good immune response without being, without multiplying in our bodies. And now I'm going to skip the DNA vaccines, but really important, and these vaccines now, the viral vector vaccines. This is basically the Oxford AstraZeneca vaccine, where we use a kind of a virus capsid, an adenovirus capsid as a delivery kind of capsid, and inside we have DNA, and the DNA codes for the spike protein, yeah, for this protein here. And when we get this delivered into our body, our body makes this kind of spike protein, and then it can basically make antibodies against the spike protein in our body. And something similar, not the same, but similar, is going on with the mRNA vaccines. So these are kind of nucleotides, kind of RNA vaccines. So this is a piece of RNA here, coding for the spike protein again, and it's encapsulated in a little lipid particle, and this is also delivered into our body, and then the spike protein from the coronavirus is made inside our body, and then we can make antibodies against the spike protein. Again, this cannot multiply in our bodies, and this viral vector can also not multiply. We just give it, the spike is made, and the antibodies can be made. So on the right-hand side of the slide here, you can see the Pfizer and Moderna vaccines were our mRNA vaccines that are currently on the market against COVID-19, and the viral vector vaccines based on adenovirus, the Oxford AstraZeneca vaccine, the Sputnik, and the Janssen vaccines. So each, as I said this earlier, each of the vaccines requires kind of a different manufacturing process. And this is where the biochemical engineers are coming in again. Yes, I'm coming back to biochemical engineers. So we have different types of organisms where we make the vaccines in. So bacteria, yeast, or mammalian cells. We give the DNA to those kind of expression hosts. This is what we call them. And then they make either a whole virus or a virus-like particle or kind of the subunit vaccine. So all these different types that we've seen here can be made by these kind of hosts, yeah, our kind of workhorses, if you want. And then we need to decide how do we want to get these products out of these cells? What kind of purification methods are we using? So as a biochemical engineer, you will also kind of design this process. How do I culture those cells? How can I purify this away from the rest of the cell components? How can I make a safe product? And our goal is always to increase our speed, how as quickly as possible react if we have a pandemic or an outbreak. So bring a vaccine to market, secure supply so everybody has the vaccine in the world, reduce risks of another kind of threat, and have a rapid response. And one approach to this is to generate so-called technologies that are platform technologies, like we have seen for the mRNA. Sorry, jumping back, we've seen for mRNA and viral vector vaccines. These are platform technologies, yeah, where you can take, let's say, three different viruses. Theoretically, in the future, you know the DNA of the kind of the protein that sits on the surface here. The code, the genetic code. You can plug them into you can make mRNA of all of them. You produce them in the same way with the same manufacturing process, but you can make essentially for each of them a vaccine that is kind of being made in the same way. The only difference is the coding sequence is different between the vaccine types. So this is our goal in the future, to be faster and less diverse in terms of how we make vaccine, to reduce the costs and react to pandemics. And just to give you a flavor of how much work goes into vaccines. For instance, for COVID-19 vaccine development, when I looked this data up, it was a few months ago, we had 132 vaccines in clinical development going through the clinical trials, and 194 different types of vaccine in preclinical trials. And all of the ones that are on the market are now either viral vector vaccines, or inactivated virus, or RNA vaccines, or protein subunit. But there's loads of other candidates that are being developed right now. So just wanted to show you that the Oxford AstraZeneca vaccine has been made, kind of what the process was for this. The group in the Jenner Institute took a chimpanzee adenovirus. This is a very safe virus because it's not, we're not normally, we don't encounter this. It's an adenovirus. We encounter other adenoviruses. And this was modified to be unable to cause any disease, so can't cause any disease. And then into the genetic sequence of this, they took the spike protein sequence, DNA sequence. We know this, and we can then generate a adenovirus with the kind of spike coding for the spike protein. And then this gets put into, basically gets multiplied in certain kind of mammalian cells, and then we purify this very high purity, and then the vaccine is given to us and then reproduce from this adenovirus, the spike protein, and then we make the spike protein, and then our immune system makes antibodies, and next time the COVID-19, next time we're infected by the coronavirus, we have antibodies already, and we are immune against this pathogen. And finally, I kind of, I'm running a bit short in time, it's been talking too long, but I wanted to show you some other kind of novel technologies. For instance, you probably heard of Novavax, which is a nanoparticle vaccine. So it's different to mRNA and it's different to adenovirus. In this case, we can use moth cells. So, from the moth insect cells, and we can make the spike protein. Well, not we, but the company makes the spike protein. And then you can see the spike protein in green here gets very highly purified and gets assembled into like a little particle that looks a bit like a virus and has the kind of the size of a virus. So I'm interested personally with my researchers in those kind of nanoparticles. We call them nanoparticles because they are between 1 and 100 nanometers in size. So they're very, very small, a very small fraction of your hair. So they can be used to put like proteins like spike protein on the surface. And so we can expose so-called antigens on the surface in a very repetitive manner. And this can also be a very modular system where you can stick on the antigen of choice onto the kind of same type of nanoparticle. And the idea is that you're making something that looks a bit like a virus, is very repetitive in showing your antigens, and should be quite efficacious. And in fact, the Novavax vaccine that I've shown you here is very, very efficient. It's almost as efficient as mRNA and viral vector. Anyway, so I'm not going to go into detail because it's very, very scientific here. So I'm just showing you we can put like the spike protein on the surface of these kind of nanoparticles, and we can make we can mix and match what we're putting onto the nanoparticle. And this is kind of a novel technology that we are looking into in my lab in terms of these kind of carrier kind of nanoparticles. How can we assemble them, and how can we stick these antigens on the surface? And I had a little example here. There's some kind of research studies that you can find already in the literature where people have done this. I'm not going to go into detail. I just want to give you a flavor. They use like a little ferritin particle, which is a natural particle, to put like a spike protein on the outside, very repetitive display, and then they purified this kind of particle using kind of a shake flask, culturing in cells, and then the purification via some chromatography columns. And then this product gets put into like a little mouse, like for mouse studies. And then you can check, did they make antibodies? How many antibodies did they make? How long did it take? And so on. So I'm not going to go into detail. It doesn't matter. I just want to give you that little flavor. So in my lab, we work on these kind of different types of particles that look like virus-like particles. They're not all virus-like particles. They're different protein particles from nature that we are engineering, and that, as you know, biochemical engineers that come into my lab, like undergraduate project students and PhD students, they do work on these kind of structures. And we also starting with my colleagues, we collaborate with my colleagues to develop these kind of different processes to to work out how to better make our particles, and in the future, hopefully, to make nice platforms, modular platforms for fast development of vaccines for future threats, and hopefully not pandemics, but future outbreaks. And with this, I'm sorry for talking quite a long time. With this, I'm going to end. Oops, sorry. I'm going to end my presentation. Let me come back here. I don't know what happened. And I'm passing back to Kim for questions.
Fantastic. Thank you very much. So I'm just checking if I can see. Sorry, thank you for the kind words in the chat. Somebody said some very kind words about the presentation. We appreciate that. So we've had a few questions come in. Just to remind you, please put your questions in the Q&A box. We're really looking forward to getting through them. You can post them anonymously if you wish. You can just click a little button and that's it. They're asked anonymously. Some really, really good questions coming in. I'm also going to put a link to the UCL undergraduate open days. Having seen the composition of people from the polls that you filled in earlier, I can see that there may be a few prospective undergraduate students. So we're doing a Q&A presentation tomorrow with the admissions tutor, Dr. Jack Jeffries, tomorrow at 5 o'clock, and there's one on Friday at 9 o'clock. So whichever one works for you, if you finish school in the UK or if that works for another time, they'll be the same session but repeated at different time slots. You'll also be able to talk to one of our students who'll be there, so you can ask them, and they'll be devastatingly honest. They always are. Anything you want to ask them about studying in UCL Biochemical Engineering or UCL generally. But hopefully, Steffi will have a chance to look through the questions. What I'm going to do is I'm going to start off by asking this one flat out to Steffi. Steffi, in real life, is it as interesting as it sounds?
Absolutely. Yes, it is super interesting. I think we see this as well, Kim, when we give this undergraduate module on vaccine. Obviously, there's a lot of interest from our students, and I think we do need this next generation of biochemical engineers going into this field, because we see now, after this pandemic and even before this, how important it is that we keep moving forward and really develop, not just develop, but also look into the manufacturing and how we can be, how can we kind of innovate the vaccine world as well, and how can we be better at kind of distributing vaccines across the world more equally, and so on. Yeah, so there's lots of challenges, and we do need a lot of biochemical engineers to help.
Great, thank you. Steffi, have a look through the list of questions, and in case there's anything you've already addressed there, or you want to group them up, just let you know, I have put a link to the undergraduate open days happening tomorrow and Friday in the chat. Also, if you're interested in studying something postgraduate with us, we haven't got any sessions in the near future, because we've just closed, the deadlines are coming up very shortly. So we had our postgraduate open day earlier in the year. But I'll put a link in the chat. If you want to leave your details with us, we'll let you know about any events that we've got coming up. And also, I noticed there are a few people from London. If you want to come along and visit us in person, you can have a tour of the pilot plant and see the facilities in the building, meet the team. Those sessions are going to take place on the 15th and 16th of July. And you can sign up to those using the same link. That's mainly for undergraduates. But as a prospective postgraduate, if you want to come and see the building, come for a tour, there'll be this. Please come and join us in person. You can get to see us in real life. So Steffi, any of those?
Yeah. Let me read them out. So there's a question if it's necessary to develop a new vaccine for the recent emergence of monkeypox virus. Thank you for asking this. I'm not sure. I'm not a clinician, so I will try and answer this with to the best of my knowledge. Yes, we've seen the headlines recently about the monkeypox outbreak. I've seen that there was treatment with the smallpox vaccines. So it seems that we can treat with smallpox vaccines. So I guess this, I mean, it's a bit early to be honest to say this now. It depends how the situation will develop. But I think if the smallpox vaccine is very effective, then, you know, I would imagine that we can use that. Yeah. And then there's a question about for the vaccine that uses antigens from the virus, do you produce the antigen through protein synthesis using the virus RNA, or extract the antigen from the virus itself? Yeah, that's a really nice question. Thank you for asking this. So we can do both, actually. So if you think about the flu vaccine, we can make the entire virus. So this is actually what has been done traditionally. So we can grow the virus in eggs. It's a very interesting process. I didn't have time to explain, but industry grows the virus in eggs, lots and lots of eggs and batches of eggs, and then they isolate the virus from the eggs, from the chicken eggs, and then you purify very highly the virus, and you can take parts, kind of the antigens from the surface. They have very specific proteins on the surface of the influenza virus that can be taken from the surface, from the virus, and then this is formulated into the vaccine and given to us as a, you know, very standard flu vaccine. It's been around for years and years, and industry really, you know, it's a very established process, and it's also a cheap process. In terms of we can, but the other question was very good as well. So yes, we can also produce just a protein, like for example, if I come back to the flu virus, we can take one of the proteins, like the hemagglutinin protein from the surface, and we can specifically produce that protein, for instance, also in insect cells and other cell lines, and we can just purify that antigen itself and give that as a vaccine. And this is also on the market. So both are already on the market. Very good question. Okay, how long can a vaccine be useful in a person's body? Also, thank you, good question. So it depends somewhat on the immune response, on the type of vaccine and immune response. For instance, if I pick out the COVID-19 example, so if we have been given, let's say, the Oxford AstraZeneca vaccine, the trials showed that we have protection after the first dose for about 3 months. So, and then, you know, our antibodies are still very high after, you know, until after 3 months. So the second, like the booster vaccine, is then given after that 3 months to kind of give like a booster to your body and to create kind of memory cells. So first time you get the vaccine, you have your initial response, yeah, you make your antibodies and you make some memory cells. And when we give the booster, we give more of the antigen, and, you know, your body recognizes it. The memory cells will be able to make more antibody, and then you have a few more months after this, half a year or so, where you still have your antibodies that can react, and you have your kind of memory cells, yeah. But that depends very much on the vaccine, and it's very well normally observed and tested, and, you know, we know roughly how long immune response is, and then we can give boosters. This is what boosters are for, to kind of stimulate and refresh the immune response again. I hope I answered this. Are there any nanotechnology modules within the biochem engineering department? It seems to be really interesting. Yeah, in terms of the nanotechnology, yes, we do incorporate the nanotechnology type research areas within our undergraduate modules. So for instance, we have an introduction to biochemical engineering module where we touch up on, that's in the first year, touch up on various of those kind of technologies more on a kind of fundamental level, and then we build on those. For instance, in the vaccine course in the third year, then when we talk about like analytics and upstream and downstream processing, we touch upon those kind of technologies too. We do work also closely with the London Centre for Nanotechnology. So I have, for instance, some personal kind of research collaboration with Beda that work on diagnostics, so also on nanotechnologies. So yeah, you do get kind of exposure to different types of developments throughout the degree and the different modules. Yes.
Okay, I just want to quickly point out that we've got 4 minutes left to go, so we're going to pick either 2 short questions or one long question. And just to reiterate, if there's anything that we've not answered here, there are actually some sessions coming up. If you go into our website, you'll see that we've got some taster days we're running in conjunction with the University of London. So you can actually go a bit deeper into the subject, and Steffi's hosting one of those with the iGEM team, which is in July. And you can register for that on the front of our website. So if we've not gotten through it here, you can actually come along then as well.
Yes. How can vaccine packaging be made more environmentally friendly? Is it possible to make packaging which is biodegradable, or is this a biohazard? Does all waste need to be incinerated? If this is the case, is it worth even making packaging biodegradable? So this is a really nice question. Again, you're asking all these good questions. Actually, we've just had a vaccine training course for our industry delegates and we had some presentations from various kind of manufacturers, and there is some direction towards looking at storage, stability, and kind of delivery. So the packaging kind of falls within this aspect. This is why I'm mentioning it. So if we can think about more innovative delivery in the future, we think at the same time about packaging, right? So there's some ideas that we've seen about kind of delivery to the skin via some kind of little needles or patches. So these would be much, much smaller and less invasive than using an actual proper kind of surgical needle. And we wouldn't need so much glassware in the future and so on. This is all in the development as we are speaking, so it's not yet on the market, but there's definitely a drive, like you said, to to look into that and also to become much more sustainable in the future in terms of how we make the vaccines, in terms of the manufacturing, but also then the packaging and the storage. And the other thing is the kind of storability. We kind of want to avoid the cold chain. I didn't say that so much, but mRNA vaccines require very cold temperatures, so something like -80. In some instances, some of them are now okay for the fridge, but in the future, can we store them more like room temperature for longer time? Packaging will come into that too. So thank you for asking that. Okay, can you explain? Sorry, Kim,
I
did you
just
want
want
to
to say quickly, somebody's asking about the difference between bioengineering and biochemical engineering. I don't know. Just want to say, if you're interested in a degree, come along and we can tell you the difference between chemical engineering, biochemical engineering, biomedical engineering, because sometimes people get confused between this stuff, and we can go into that in a bit more detail.
Yeah, exactly. Yeah, I think that would be good if you could join one of our other sessions, because you see that biochemical engineering is more than bioengineering. We talked about bioengineering a lot today because that's more my focus, but in the department we do a lot of biochemical engineering and process engineering, really looking at the process. How can I purify? How can I make this product better? What kind of bioreactor do I need to design? We talk about disposable kind of, or sustainable ways of growing and purifying and so on. Yeah. So can you explain how viruses are grown in eggs? Unfortunately, we haven't got enough time to do that today, but there's some really good resources online if you want to look at that further. But you need basically embryonic kind of eggs, embryos, and you would you will have to inject kind of the live virus into them, and they grow, and they have to be in a certain stage of growth in those eggs, and have to be infected with the virus and then grown over several days and then harvested. So it's quite a it's a lengthy process. So I won't have much time today to explain that, unfortunately, but look online and see if you can find some more information.
So I just very, very quickly want to address one of the questions somebody's asked about about biochemical engineering being an interdisciplinary subject. after this session, it may take a couple of weeks, but you'll get a link to watch a recording, but you'll also be able to watch a recording of all the other sessions. And the one I was in last week with Dr. Rana Khalife, they were talking about, and we had a really good chat about the interdisciplinarity of the subject, and she was talking on stem cell gene therapy, manufacturing, commercialization, and innovative medicines, and how you make them and get them to market. And it's very exciting area. But my colleague in that session was talking about what it means being in a university where you're actually multidisciplinary, and where you get the intersection of not just medicine and technology, but also arts and humanities and social historical sciences, and what it means to them. So when you get the link to the recording of this, you'll also be able to watch all the other sessions back, and do watch that one, especially at the end for the Q&A session. I think you'll find it very interesting. But I'm afraid we've come to the end of our time. Thank you so much, Steffi, and thank you everyone who's joined us today. It's been great to have you here. Thank you for your brilliant questions. Once again, we're overwhelmed by how good these questions are. I'm really sorry that we just don't have enough time to go through them all. We hope to see you at one of our other events, and we really hope to see you in person at some point. So with that, thank you very much, Steffi. Thank you for your kind words, and I hope you have a great day the rest of the day.
Thank you, everybody.
Thank you very much.
Have a good day. Goodbye.